Hello everyone. So today we are going to discuss regarding tuberculosis. So if you see this tuberculosis is a bacterial disease which is caused by a bacterium called Mycobacterium tuberculosis. And lungs are the most common places or common organs which will get affected by this tuberculosis, and we'll call it as pulmonary tuberculosis. But there are several other organ systems which will get affected in case of tuberculosis, such as intestine, bones, brain, kidney. And this Mycobacterium tuberculosis, which is a causative agent for this tuberculosis, is also known as Koch bacilli. So after its discovery by Robert Koch, and he is that great personality who discovered this Koch bacilli, Mycobacterium tuberculosis, which is a causative agent for Mycobacterium tuberculosis. Tuberculosis. And these are all the different eminent personalities who suffered from tuberculosis and who died of tuberculosis. And this is Srinivas Ramanujan, and this is Kamala Nehru. And Mycobacterium tuberculosis is an aerobic bacterium. Mostly it will reside on the apical regions of lungs where the oxygen availability is more. So infection of the lungs by Mycobacterium tuberculosis, we already said it is known as pulmonary tuberculosis. And sometimes we will also observe this tuberculosis will get disseminated from lungs into the intestine and causes intestinal tuberculosis, and to the brain so whenever it disseminates to the brain, we will call it as meningeal tuberculosis. Coming to the mode of transmission, and the mode of transmission, the most common mode of transmission of this tuberculosis from one person to another person is inhalation. We already said this is a communicable disease which spreads from one place, so one person to another person. So inhalation is the most common route of transmission. Whenever an individual who is getting suffered from tuberculosis, if he coughs or sneezes, so at that time, whatever the cough droplets which are there, so in those cough droplets, this Mycobacterium tuberculosis will present, and so it will transfer. So it will get entered into the environment, and those who will inhale those Mycobacterium tuberculosis contain those cough droplets, so they will also develop tuberculosis. And so development of tuberculosis upon inhalation of those cup droplets which contains that Mycobacterium tuberculosis depends on some parameters. It depends on the immune status of the individual who inhaled those droplets. If he is having a good immune system, he will develop a poor that means very mild symptoms of tuberculosis. And if he is having a very poor immune system, he will develop tuberculosis in a very severe, so very early, as well as we will observe very severe symptoms. And another parameter on which this development of tuberculosis depends on a number of bacilli released into the air while coughing, and environmental conditions like humidity in the air. Humidity will enhance the process, that means which will enhances the development of tuberculosis, and proximity, duration and frequency of exposure to those cough droplets, and presence of other diseases like HIV and AIDS. All these are all the parameters which has to be considered. All these are all the parameters which has to be considered whenever whenever we have to suscept an individual, whenever he inhales the cough droplets of an infected tuberculosis individual. And this is a picture representing the transmission of tuberculosis through inhalation route. So whenever the infected individual coughs, whatever the Mycobacterium tuberculosis which are present in his lungs will through his mouth, so will move through his mouth and in the form of cough droplets, and those cough droplets will be inhaled by another uninfected individual. So and he develops Mycobacterium. So and he develops tuberculosis. And another mode of transmission is ingestion. So whenever if any individual is getting suffered from tuberculosis, especially pulmonary tuberculosis, there is a chance that whatever the sputum containing Mycobacterium tuberculosis will be ingested by that individual. So which results in the entry of that Mycobacterium tuberculosis into his stomach as well as intestine, resulting in intestinal tuberculosis. And sometimes even even through inoculation, this tuberculosis will be transmitted from one person to another person. And so very rarely we will observe this inoculation. And tuberculosis will also transmit through the placenta, so from an infected mother to the fetus, and lead to congenital tuberculosis, and it is a very rare route of transmission. Coming to the pathogenesis of tuberculosis is a very important thing which we have to know regarding this pathogenesis. So, and it's a very interesting thing too. So tuberculosis, whatever the inflammation which is observed in case of tuberculosis is nothing but granulomatous inflammation. So whenever this Mycobacterium tuberculosis enters into your lungs, so your alveolar macrophages which are there, those alveolar macrophages will try to engulf and kill that Mycobacterium tuberculosis, but they will not cause, that means since they don't have the ability to kill that Mycobacterium tuberculosis, so in doing so, they will undergo some damage and they will change their shape as, so that means, so epithelioid cells, that means, so whenever, so while killing, that means whenever these alveolar macrophages tries to kill Mycobacterium tuberculosis, since they don't have the ability to kill that, but they will try their best, but ultimately it results in change in the shape of alveolar macrophages, which results in the formation of epithelioid cells. So since that modified macrophages represents an epithelial cell, we are calling them as epithelioid cells. And after that, so this, that means these alveolar macrophages will release some cytokines such as interleukin 12 and sends a message to the T cells. So T cells to kill this Mycobacterium tuberculosis, and these T cells, especially Th1 T helper cells, will release various number of cytokines in order to kill the bacteria. But instead of killing the bacteria, so those cytokines which were released by this T helper cells will cause us damage to the background tissue which is there, and ultimately, and as well as whatever the macrophages which are, whatever the already deformed alveolar macrophages which are there, what we are calling them as epithelioid cells, those epithelioid cells will combine each other and forms tumor giant cells. And those tumor giant cells, normal alveolar macrophages as well as T helper cells will tries to release, so they will release various number of cytokines in order to kill that bacteria, but so ultimately, so they fail. But whatever the background tissue which is there, so that background tissue will undergoes damage. So here the thing which we have to remember is, so Mycobacterium tuberculosis didn't release any exotoxin or endotoxin, but whatever the immune attack which was developed against that Mycobacterium tuberculosis caused some damage to the tissues itself. So ultimately it results in the formation of a granuloma, and since it is because of tuberculosis, so Mycobacterium tuberculosis, we'll call it as tuberculoma. So this is how this is how the granuloma formation will be there. So this granuloma comprises of alveolar macrophages, modified epithelial cells, so epithelioid cells, tumor giant cells, and as well as T helper cells. So coming to the screening test, which is a very famous screening test for tuberculosis. As we already discussed regarding leprosy, so in leprosy we discussed about tuberculin test, lepromin test, and as in the same way we have tuberculin test here, which is also known as Montauk's test as well as Pirquet test. So what we will do is, so if any individual is having the symptoms of tuberculosis and if he comes to you, so to screen whether he is having tuberculosis or not, what we will do is just we will inject a purified protein derivative of tuberculin protein of 0.1 ml intradermally in his forearm, and we will just that means observe for the development of any hypersensitivity reaction around the site of injection. And if he develops any induration at the site of infection, so based on the diameter of the induration, we will decide whether the Montauk's test or the tuberculin test is positive. So if the induration is of greater than 9 mm, if the induration is greater than 9 mm, so we will say the Montauk's test is positive, or otherwise he is a past previous TB patient. And if the induration is 6 to 9 mm, so we will say Montauk's test is doubtful, and the induration is greater than 6 mm, we will say that Montauk's test is negative. So sometimes, and this Montauk's test or tuberculin test is just a screening test, is not a confirmatory test for the diagnosis of tuberculosis. Sometimes this Montauk's test will give you a false positive result, and sometimes it will give you a false negative result. So false positive, whatever the positive result which was given by this Montauk's test is false. False negative, whatever the negative result which was given by this Montauk's test is false. Sometimes it will give false positive. So even though the patient is not having tuberculosis, but it will give a result of positive. Even though so sometimes it will give even though the patient is having tuberculosis, it will give a negative result. So these are all the different conditions where we'll get a false positive result. So the first thing is if we use a faulty technique of injection, that means if we are not doing that purifier, that means injecting that purified protein derivative of tuberculin protein so into intradermal route, then we may get a false positive result. Or if there is any degraded tuberculin protein in that, we will get a false positive result. Or if you give a very deep injection, or if you if you go with a repeated tuberculin test, or if the individual previously vaccinated with BCG, so then we will get this false positive result. In pre-allergic phase and high fever, measles, chickenpox, whooping cough, malnutrition, HIV or AIDS, usage of anti-allergic and immunosuppressant, in these conditions we will get a false negative result. And you all know BCG vaccination. So as the vaccine which was given in order to prevent tuberculosis, generally the dose of this BCG vaccination is 0.1 ml. So generally it is given in 0.1 mg in 0.1 ml intradermally. Generally it is given skin over left deltoid muscle. So this is the BCG vaccination, and you can, so I think all the people 90% or 95% of the individuals who are Indian residents will receive this will receive this BCG vaccination in their left arm deltoid muscle. We can observe this mark lesion. So in each and every individual who was vaccinated with BCG. And if you observe the symptoms of tuberculosis, so the most common symptoms of tuberculosis are nothing but cough. Sometimes we will also observe cough with blood, and the sputum. So that means rust-colored sputum is the most important thing which we have to remember in case of tuberculosis. In case of tuberculosis, we'll observe sputum. Sputum will be formed most commonly, and that sputum will be in rust-colored. We'll observe chest pain as well as weight loss. So weight loss is due to loss of appetite. Those individuals who are getting suffered from tuberculosis, they will have very less appetite. So because of that, so they won't eat anything and they will become very lean. And we will observe tiredness, fever, night sweating, chills, and so not feeling hungry, which we discussed as loss of appetite. So these are all the different symptoms which we will observe in case of tuberculosis. And sometimes this tuberculosis will also affect the other parts, as we discussed earlier. It will affect the kidney, spine, brain, if even intestine. So if it causes, so intestine, we'll call it as intestinal tuberculosis. If it causes brain, we'll call it as meningeal tuberculosis. And even it may also affect the spine, resulting in spinal tuberculosis. And even if it affects the bones, we'll call it as bony tuberculosis. And the diagnostic test includes chest X-ray. So whatever the Montauk's test or tuberculin test or pipette test which we discussed is just a screening test. Screening tests are not diagnostic tests, so they are not confirmatory. Just it will give a idea whether the patient can be gone. So that means whether the patient can be forwarded to the diagnostic test or not. So if you see this diagnostic test, most commonly the TB will be diagnosed by chest X-ray. And after this test, a very confirmatory test which is there. So confirmatory test will be done through sputum smear examination. Just they will take the sample of sputum from the suspected individual and they will observe it under microscope for the presence of this Mycobacterium tuberculosis, and by culture of this mycobacterial species which is there in the sputum. So these are all the three diagnostic tests which are used to diagnose tuberculosis. Thank you.